I'm very happy to present to you our speaker of today. It's Professor Dr. Florian Gantner. He is currently head of Translational Medicine and Clinical Pharmacology at Boehringer in Ingelheim. And he had a number of positions also in academia. He was he's honorary professor of the University of Manchester and was lecturer at University of Duisburg, and had a career with a number of companies. So he's very knowledgeable in the requirements and the methods and the pitfalls and the successes of translation. And therefore, without further ado, I would ask Florian Gantner to start his lecture, and I'm very enthusiastic to hear what he has to tell us about his view on translation.
So this is the outline of the talk. I go right into things. I hope I can provide also not only the frontiers, but also some successes, both with regard to the scientific frontiers, but also with regard to the structural challenges that we see in the context of translational medicine. Now, what do we understand under translational medicine in the context of drug development? Basically, this is the integrated application of clinical methods and technologies, including biomarkers of many kinds and flavors, and I'll come to that later, pharmacometrics approaches in terms of modeling and simulation, etc. And all that in order to improve our confidence in our new drug candidates. We want to understand better the risk-benefit of new molecules, in other words, the therapeutic index, and want to come to more and better, earlier, cost-effective decisions by proving that a new mode of action does work or doesn't work in man as early as possible. And we refer to that milestone of clinical development as a proof of clinical principle, and you will hear this type of terms in many different definitions, but this is the one that we use in the company I work for, which is BI. And of course, translational medicine is also important in the later stages of development, and that is during the confirmatory phase. And all that, of course, is to, for us as an industry player, to provide a competitive pipeline in the areas we are active in. And of course, and that's the center of everything we do, to provide new and hopefully effective medication to patients in need. Now, if we look at the pharmaceutical R&D value chain, and I'm sure all of you are familiar with this, starting at the left end with the discovery phase up to registration of a new compound, and then post-launch and post-marketing production, some additional expansions of the label, for instance, or of indications. This normally takes us about a decade. And the big dilemma that we have in pharma R&D is summarized in 3 words. It's very lengthy, it's costly, and we have low success rates. So all combined, not very good. And here is what we hope translation medicine, with what I outlined before, can help quite a deal. And in my 2 to 3 decades of working both in drug discovery and in early clinical development, 2 dominant questions have always repeatedly come up, and basically in every project. Number 1, will hitting the target that we want to address in a given disease truly translate into the therapeutic improvement we hope for? And secondly, and more importantly, which of the patients we have in mind will respond best, and how can we identify them? And if you look for man, when starting with a molecular target and the design of a molecule that modulates that target through a lengthy investigation in vitro and in vivo, we hopefully then come to the clinical efficacy that we hope for. And this is depicted here with these half arrows in both directions, not a linear, but a rather imperative and step-by-step feedback cycle in all these phases. And 2 learnings in the 2 to 3 decades I've been working in this field demonstrate that this is all but a straightforward dynamics analysis. Because mice turned out not to be just furry plastic plates, and everybody who tries to translate something into a mouse from an in vitro experiment knows what I talk about. And secondly, the step from rodents to man is also huge, because humans are no nude mice, as we all know. Now here the same picture with regard to the value chain, and depicted below in the various phases are where translational medicine comes into play with in our company biomarker teams that are established fairly early, already during the discovery phase, which then are complemented by analysis if we go into man and prepare for man application for pharmacokinetics and pharmacodynamics analysis. And we of course want to translate biomarkers from the animal studies into man. And in the first in-man trials, normally conducted, as you all know, in healthy volunteers, we assess safety and tolerability, but also start exploring biomarkers and hypothesis testing as early as possible in order to come up with patient populations we want to study our new compounds in first, in these, as I introduced the term before, proof of clinical principle studies. And if the mode of action and the indication allows, maybe the development of a companion diagnostics is also important. Now, moving on. To sum up, what we actually would like to provide, if you plot time and money investments at the x-axis, along the x-axis, and knowledge gain on the y-axis, you see the dotted line, which is basically, or historically has been, the knowledge gain at the end of phase 2b, and then of course through phase 3. And with the appropriate application of translational medicine disciplines and knowledge, we hope that we can bend this curve and move this curve very much to the left, to serve two purposes: stepping out if things don't seem to work, and investing in those that bear the highest likelihood of success. Now briefly to the biomarkers and their definitions. Again, biomarkers come in many flavors, but there is one official definition which is shown here. That is a characteristic which is objectively measured and evaluated as an indicator exploring biomarkers or pathogenic processes or pharmacological responses to a therapeutic intervention. This is fairly broad, as you can appreciate, and the biomarkers come in many flavors. I consider the classification by quote-unquote nature in terms of classical biomarkers, such as soluble markers you can measure in whatever matrix you wish, including blood cell analysis, etc., or functional tests. Number two, genomic biomarkers that comprise all genomic readouts you can think of, from DNA sequencing up to microRNA analysis. And then very important and very much being developed currently in many areas is imaging biomarkers. And I'll show you a few examples later. Now, when it comes to clinical drug development and biomarker applications, basically there are always questions that are being asked. And I'm not going through this list, but we divide them, and that again is a BI terminology, into three major categories. Biomarkers that tell us about the pharmacodynamics. In other words, does the molecule truly what it is meant to do? Secondly, safety biomarkers that tell us whether a compound is safe and tolerable. And thirdly, patient selection biomarkers, in order to be able not only to include the right patients in the clinical trials, but also to develop, of course, the molecule for the right patient population or patient populations. And I would like to give you two examples during the course of this talk. One on pharmacodynamic biomarkers we have applied early on, including some genetic biomarkers that helped us to profile an antibody against IL-23, now known as risankizumab, in psoriasis. And secondly, spesolimab, another antibody directed against IL-36 receptor, also used or to be developed in disease, where we look for specific patient populations that might tell us early on whether it's worthwhile investing or not. And here again is the biomarker categories that we apply in our Boehringer Ingelheim language across the entire portfolio that we develop. And these are the three categories with a few subcategories that I don't need to go into today, but that is a summary of what we actually use as a guidance to apply biomarkers during our clinical development project. Now, our colleagues from Pfizer have come out with a paper discussion a couple of years ago, which is still, I believe, very true, and which is why I brought it in, where they call out for 3 pillars of survival of a molecule during phase 2. And highlighted in yellow here are those 3 pillars: exposure at the site of action over desired period of time, binding to the pharmacological target, and then also expressing the functional pharmacological activity. So basically an appropriate PK/PD profile with the appropriate PD profile. This is what seems to be the way to a successful clinical development. And I stress this point because you might or might not know that in the 80s or 90s, when I started in pharmaceutical industry, the major reason for drugs not going forward was intolerability or safety concerns. And that has actually, not only that, and the second major reason was inappropriate pharmacokinetics and exposure. And this has changed now to the major reason of non-success being lack of efficacy, which means we normally have the appropriate exposure and tolerability and safety, but often lack the pharmacological activity that we hope for. And or we don't understand our diseases well enough to develop the appropriate new therapeutic concept for. Let's switch gears now to a first example, which is very much a trendy disease, if I may say, which is NASH or non-alcoholic steatohepatitis. This is a liver disease of high unmet medical need, where basically the call for biomarkers to make clinical decisions is very high. Why is that? This is because the grading of NASH solely still depends on liver biopsies, which has quite some limitations. I'll come to that. And the fundamental question that nobody can answer right now is, which of the NASH patients are progressing fast, and what are biomarkers that could tell us who of the patient segments would progress faster than others. Therefore, we would like to provide looks for new biomarkers to assess NASH and liver fibrosis. As I said, biopsy is still the gold standard, but the lesions in the liver, as indicated in this figure to the right, are unevenly distributed. So, and only a small region is assessed by a biopsy. Therefore, many times sampling errors lead to misdiagnosis or misgrading of liver disease. And what we would like and others to develop here is to look for alternative imaging modalities and also soluble biomarkers to establish the link to clinical NASH grading. To give you a few examples, magnetic resonance elastography bears quite some hope here to be capable of staging liver disease, and that goes over a much larger region of a liver as opposed to biopsy. And transient elastography also might help us to lead to new stagings. In particular, if these are combined with other elements listed here under the specialized biomarkers that could perhaps tell us how much inflammation is going on at what stage and in which patients, how much fibrosis already has kicked in, and how can we describe that, and how much of hepatocyte function has been lost or is going to be lost. And therefore there is a number of intents currently in consortia fashion, but also of course in individual trials to sort out which could help us to come to another clinical NASH grading to guide also drug development in this disease where not much, if anything, is available. A second example how to apply new biomarkers is summarized here in the context of immune oncology. If you look at the cycle here, lent from Jen and Melman as published in Immunity 2013, there is a number of steps where, and this is always framed in green, imaging modalities and imaging application could come into play to help us develop biomarkers for these various steps in the cycle. At the right top end of this slide, starting with dynamic contrast enhanced MRI to assess tumor, for instance, if you want to develop an oncolytic virus or T-cell engager antibodies. In terms of tumor microenvironment modulators, imaging of tumor infiltrating leukocytes and truly showing whether or not they're being present and how a given molecule is modulating it could help us if we develop checkpoint inhibitors or again T-cell engagers or the like. Of course, biodistribution studies to support novel drug formats. Many of you might know that many formats in terms of new biological entities, antibody-like molecules, dual antibodies, etc., are being tested currently and developed. They require special biodistribution methodologies and so on and so forth. Of course, tumor cell death could be looked at via a release of antigens. Again, if tumor cell metabolism and the texture is being measured, for instance, by PET CTs, and so on and so forth. So a number of applications of imaging modalities that could help us going forward. And here is summarized with two examples of active collaborations that we have partnered up with at BI in order to truly look for activated T cells and to look for specifically CD8 plus T cells via zirconium labeled anti-CD8 disciplines and knowledge we have with PET CT imaging. And that is currently being applied in some of our early oncology studies in order to learn earlier and quicker whether or not our molecules do what we hope they do. Now, I would like to come back to the first of the examples I alluded to before, and this is on the genomic biomarker example in psoriasis. We have developed an antibody directed against IL-23, a cytokine that we believe, and many believe, is actually doing quite a number of things in inflamed skin. And what we did here is we looked at the modulation of a number of genes in one of the 5 listed areas on the right hand of this slide, on the IL-23, IL-17 cytokine axis, epidermal development, keratinocyte differentiation, epidermal cell differentiation, and keratinization. And what you see here on this heat map and the numbers and the gene names on the right end, all these genes belong to one of the 5 areas where we think where we thought an anti-IL-23 inhibitor would have an impact. And indeed, this was the case if you look at the panel at the very low end of this graph. In my into work and investing in samples of patients, and every column of this heat map is representing one skin sample gene profiled of a psoriatic patient. Red means highly active, blue means inactive or downregulated. The placebo group are the ones on the right end of this heat map, labeled in green below. And as you easily can appreciate, skin samples of psoriatic patients had high activity in all 5 of these areas. However, if these patients have been pre-treated with the anti-IL-23 antibody risankizumab, most, not all, but most of them had a dramatic downregulation of genes of interest that fall into that category of these 5 signaling pathways, which brought us then to test further and really look for skin effects in patients with a clinical readout. And that is summarized here on the next slide. You see here pre-treatment a skin patient, and after treatment of a single antibody shot of risankizumab, even 24 weeks after that very single dose, the skin was still close to be clear. So very, very dramatic and very, very convincing results in these first studies. And some patients actually, they responded for more than a year to a single shot of risankizumab. If we then compare how many patients reached an almost clear, which means PASI 90, PASI stands for Psoriasis Activity Score Index, 90%, or a clear skin, which would be PASI 100, in those cohorts that we tested, which were placebo, a 0.25 mg per k or a 1 mg per k dose in patients. These were again the results. No responders in the placebo group, already quite dramatic responses, and even better responses in the 1 mg per k dose group. And that finally then after of course going forward in testing it in phase 2b and phase 3 led then to the launch 1.5 years ago, or close to 1.5 years ago, of risankizumab with the trade name Skyrizi, which is provided by somebody else but us, but that's a different story. Now, the second example I have is again a story in skin. Following the hypothesis and comparing skin to intestine with inflammation disorders, illustrated here in the middle by barrier damage leading to inflammation, which in turn leads to aberrant tissue remodeling and fibrosis, brought us to a receptor ligand system called interleukin 36. And we hypothesized that by intervening with this receptor signaling, we could also bring benefit to patients suffering from chronic inflammatory skin diseases. As indicated on the next slide, the hypothesis was to by suppressing IL-36 receptor activation, to reduce epithelial cell or fibroblast and immune cell mediated inflammation, and by interrupting the inflammatory response that amplifies pathogenic cytokine production, would bring relief to patients suffering from skin inflammation, where we believed IL-36 plays a crucial role. And in order to test this, and this brings me to the biomarker of patient selection, we looked for an indication where we had highest hopes that this might help us to answer the question whether or not this is truly the case. And we tested this in a rare disease called generalized pustular psoriasis, which is orphan, as it's very rare. It has acute flares with pustules, accompanied often with erythema and scaling, and indicated on the right end, you see some of these poor patients suffering from that very dramatic skin disease. And sometimes this is even life-threatening. But there, and that was the link for us, is a human genetic link that is available, because some of the most severely affected GPP patients actually carry a loss of function mutation in a natural antagonist of the IL-36 receptor. So obviously in these patients, the brake that helps them dealing with the inflammation is no longer available, and therefore they develop this very dramatic expression of GPP. So we thought if we could study our new molecule, which is an antibody to the IL-36 receptor, we might have highest hopes to prove or disprove our hypothesis. And this was not easy because there weren't many patients available, but we partnered up with a few centers that have had access to GPP patients. And we started basically an open label phase 2A study in a number of these patients. And we looked for moderate disease. This disease is also based on severity grades broken down into almost clear mild disease, moderate and severe disease. And we started off with studying a few of these patients suffering from moderate disease. And what happened there is the following. Here you see somehow increased in size how these skins look like. And already 1 week post-treatment with an antibody directed against the IL-36 receptor, we saw quite some dramatic changes back from moderate to mild disease, and even further down the road to almost clear skin 4 weeks after treatment. Again, very much increasing our belief in the molecule. And the data of this first study was already published also. We had 7 patients studied, all of them responded, and 5 of them were clear within 1 week's time. And 4 of these 7 patients actually were carriers of the loss of function mutation. But it also told us that there is hope for non-carriers of the mutation, and that the molecule might also work in a broader GPP population. And phase 3 studies are currently ongoing. So this is a molecule that is still in development. And we also have, of course, expanded to other diseases where we also see connections to the IL-36 pathway, such as palmoplantar pustulosis, Crohn's disease, or atopic dermatitis. Now, this is basically summing up the part of the science, and I'm not going over this slide here also to gain back a little time. And I would now like to turn it to the structural frontiers and actually share with you some experiences on 3 themes. One is global data protection regulation, which came into effect last year, as you all, I guess, know. That, of course, had implication on what we do, in particular in terms of which types of data can we work on and can we handle, and are we allowed to use. And that links into access to and use of human biospecimens, which are very important. And finally, I would like to share with you our open innovation at BI with the example of OpenMe.com, a platform that I will introduce to you. So as you all know, the GDPR basically provided for us the dilemma that health data fall into a special category of personal data, and these special categories of personal data per default should not be used. However, and luckily, there are some ways out because paragraph 1 is indicated here, shall not apply if the use of these data is necessary for medical diagnosis, provision of health in the interest of public health, etc., etc. So if appropriately applied and interpreted, there are ways out of this regulation. And this is in particular important because we not only want to create data, we also want to sample human biospecimen, which help us a great deal in drug development. For many, many reasons, and 8 of them are listed here, but there is certainly more. For instance, we can address questions from authorities without starting a new trial. If we have a very well-ordered sample bank that we can go and pick biospecimen for to do some analysis. We can, of course, also test hypotheses by going back to these collected samples. We can hopefully also enable collaborations by sharing these bank samples with collaboration partners of interest. We can confirm biomarker results in an unselected cohorts and retrospectively to test hypotheses, and many different things. So this is very important to be capable of and allowed to do. And finally, biobanking at BI has a fairly long tradition. For more than a decade, we actually sample DNAs of every of the participants of our clinical trials, and we have now started to expand this into additional biospecimen with help of partners that have subsidiaries across the globe, so that we can basically bank samples from all trials, regardless of where they are conducted. And this is also fully supported and reflected in our corporate policy on the responsible use of human biospecimen and associated data. Now, in the final few minutes, I would like to introduce to you OpenMe.com, which is an open innovation portal that BI provides since a couple of years. Now, what is this? Basically, OpenMe.com gives scientists free functional tests and preclinical tool compounds from our library. These are compounds that are highly selective for kinases, for GCPRs, for what have you, but for one or the other reason have not been developed. Either because the number 2 genomic biomarkers that toxic in some species, or because they had a drug-drug interaction profile that prohibited it from being further developed, or the like. But they are very fine compounds because they're very selective, and as tool compounds could be of great benefit. And we want to share them openly without any intellectual property or anything like that, free of charge, in our platform, which is referred to as molecules to order. We have other molecules that we would like to give out for collaborations, where we perhaps do not understand the biology well enough, but still would be interested in these molecules. And we actually invite scientists to submit research proposals in order to kind of explore jointly research areas that we share. And this has been started a few years ago, and this is already a little updated. More than 1,000 biologists have been registered across the globe. We have already given away more than 5 compounds that are being shipped after 5 clicks, and you can try it out by just typing into your browser openme.com. And everything you can register there, and everything is self-explanatory. And we have shipped already many compounds away. We have also already received a number of proposals for molecules for collaborations, and actually one success story here. One of these collaboration partners shed some light on some biology, helped us then taking back this molecule into our own research and development portfolio. So here again is the summary for the open platform that we provide: molecules for collaboration, molecules to order, and very lately, OpenToExperts. And OpenToExperts is actually to invite scientists across the globe to help us answering very specific questions that our scientists pose on that platform. You can have a look there as well. Back to the compounds order, here are just a few examples of molecules. They fall into many categories of highly selective molecules that we give away for academic partners to use them in their studies. Or here, molecules for collaborations in the field of SGLT6, for instance, or adeno-associated viruses, BCL6, SOS1, KRAS, etc. Across the many therapeutic areas that BI has interest in. And to sum up here, the OpenToExperts is basically on the background of crowdsourcing, because as you all know, crowdsourcing has shown that the output is much higher. Also, if expert questions are being addressed by non-experts and by experts of other areas, we want actually to capitalize on that, and the process is fairly straightforward. Also listed on the platform are very specific questions, and everybody who can contribute in answering is invited to contribute there. So it's very fast, flexible, and dedicated to support innovation. And with that, I'd like to sum it up here. The scientific frontiers I see is still sometimes lack of disease understanding and lack of understanding of our drug candidates, and limitations in applying biomarkers appropriately. And in terms of the structural frontiers, I shed some light on the data protection regulation and how we deal with that, including the usage of human biospecimen. And the interests of industry and academia sometimes differ, which often has been limiting. But with efforts like OpenMe, we hope we can also improve that situation. And with that, I stop here. Again, sorry for the technical problems at the beginning, but I'm very happy to take your questions now. Thank you very much.
Yeah, thank you very much from our side, and for this very interesting and thought-provoking presentation. And I do invite more questions from the audience. There's one to start from the biomarker field, and Ralf Meckle from the Center for Stroke Research is asking, has the magnetic resonance spectroscopy and/or magnetic spectroscopic imaging for the liver been attempted to use as a biomarker for NASH?
Thank you for that question. That is exactly what we want to apply in our upcoming studies. To my knowledge, there have been a few studies out which point into the direction that this is going to potentially be successful, as some comparisons are out there where the grading of liver disease has been compared by applying the traditional biopsy-based gradings and the associated with the clinical readouts, and with the new elastography or with the magnetic resonance methodology. But to my knowledge, no truly reliable study with a new molecule that should modulate the progression of disease.
Okay, so if I may follow up on that, with respect to imaging, you were stressing the importance of imaging as a additional area to generate biomarker information.
Yeah.
Do you think that is properly developed? Because you are also alluding to the interaction between academia and industry. So how would you perceive imaging to be pushed forward? Do you think that this is well enough done by academic centers developing their imaging, or do you rather envisage industry labs for imaging, or should there be joint efforts to push imaging?
Yeah, maybe a mix. I see, of course, clinical centers where imaging methodology is being applied and developed in the lead. But what I have in mind is to combine actually three partners, if you wish, the two that we just mentioned, and then complemented by the application of artificial intelligence and machine learning to deal with the many pictures that are being taken and assessed. And there, I think, is a lot to be learned and gained. This certainly requires time. Some areas are a bit better. You certainly know that in the detection of aberrant cancer-like structures, machine learning is being applied already. So oncology pathology is fairly advanced. When it comes, for instance, to diseases such as liver or the lung, we are far from it. But I think there is a lot to be gained in these areas, which is why we need to invest appropriately. And I think this calls for partnerships.
Okay, probably also with the technological industry developing the machinery around it.
Right.
Yeah.
And
so I have a few more questions here. One is from Oliver Klein from the BIH Center for Regenerative Therapies. He's asking, besides the genomic biomarker, how promising are other molecules, protein isoforms, metabolites, lipids, that can correlate closer to the phenotype for patient stratification?
Yeah, basically, I am very agnostic with regard to which kind of class a biomarker falls into, as long as that biomarker candidate is capable of answering a question. And if it is an aberrant form of a molecule or a special isoform of a protein, if it serves the purpose and if we have the analytical methods that allow us to apply this during clinical trials, then I'm fine with it. So I wouldn't necessarily rank genomic biomarkers higher than classicals or higher than imaging or vice versa. I think we have to take what is offering the highest promise in a given context of a question that we want to answer. That's basically it. So I would certainly consider them if they are of help.
And how do they relate to the role of genomics in current biomarker analyses?
I'm not sure I get the question correctly, but if...
Well, I read the question, to what extent do these molecules play a role as potential biomarkers today in comparison to genomics?
Okay. Well, I personally, out of the own portfolio that I oversee, I can't come up with a let's say modulated protein that has been used, but I'm certain there are examples out there which I would need to search for, frankly speaking. So I can't come up with a concrete example top of my head. I'm sorry for that.
Then I have another question by Regina Stark, also from the BCRT. Which tools does BI use in preclinical development to improve translation rate next to biomarkers, like organoids or organ-on-a-chip approaches?
Yes. So all of the mentioned approaches are being used. We have quite extensive collaborations going on with organoid usage in the field of oncology research. We also have started collaborations on kind of mini brain explants, if you wish, or organoids. Chip technologies are being applied in many areas. So basically, we do, again, if they fit to the question to be answered, we are open, and we do use some of them already since quite some years.
Then I have a last question here, which comes from a different angle, from the BIH Health Accelerator. And the question is, how does BI use AI in order to find out the most promising compounds or the best matching patients? So how about the use of AI in your company?
Thank you for that question. Also here we have started a number of initiatives. You may or may not know that about 5 years ago, we have started a company in the company called BIX, where we have hired data scientists capable of these analytical skills. And we have also started a number of initiatives in the digital space to help us in exactly what you referred to. And for instance, one program that we have internally set up in the field of chemistry, medicinal chemistry, is actually to use artificial intelligence to make suggestions for synthesis of molecules under the consideration of drug-drug interaction potentials, toxicological elements and fragments that shall be avoided, etc., etc. And the first results of these suggested synthesis campaigns actually have been surprisingly positive, I have to say. So we can spare, I hope, a lot of medicinal chemistry labs in the future by applying these type of technologies to help us designing quicker, better molecules. Just to give one example, we have another digital program running, which is helping us to identify patient populations based on biomarkers. And this is actually just being rolled out across the entire collection of clinical data repositories in the BI world, but we would also then, after that is done, want to expand to external healthcare databases. So there's a lot going on in this field.
So that's good news for the computer people, but less good news for the wet lab people. but there are always
no. I have to object. You always need very clever medicinal chemists, biologists, etc., etc. And when I say we can spare some medicinal chemistry labs, I'm sure we have enough other work for them.
I was just teasing, so thank you very much again. And I think there are lots of things to follow up, like you were saying. OpenMe is an offering by BI to the academic world, but I would also be interested to know which kind of offerings of the academic world in the future you would want to see, so that we could follow up on this discussion to strengthen our mutual cooperation, because I think in translation many partners are needed, and they need to work together, because not a single
Now I lost you. Whatever
unit can work on the whole value chain from the early. So do you have out of the hat a request to the academic world, what I would want from the academic world, other than cooperating in the usual way? So where do you see the deficits of our offerings?
Yeah, I think what has been a limiting factor is that sometimes the let's say the expectation wasn't managed. The mutual expectations were not always managed from the very beginning in the right fashion, which sometimes then led to failures of planned collaborations, and mostly they are not scientific, because my experience is the scientists are always fairly quickly aligned on what the questions are to be addressed. But when it comes to the framework of the contracts, of the rights that might or might not for inventions that might or might not come out of these type of collaborations, then often things have been, in my view, too complicated, and that often didn't lead to the most fruitful collaboration attempts. Yeah, just to name that. So a little more, let's say, openness in sharing, and that goes also against pharma for sure. From both partners, I think would help us quite a bit.
And do you see a difference in universities or academic institutions which have a more professional innovation or technology transfer unit, so they are more used?
No, not necessarily. Not necessarily. They sometimes even complicate, in my experience. But a trend that I see, which is I think going in the right direction, the openness of sharing data is increasing on both ends. This is reflected by sharing of registries, sharing of public databases that can be purchased or actually looked at for free. And if this is a trend that will continue, then I think many more partnerships will be the result. And okay. I'm looking forward to that.
I think that's a very good closing remark, and we hope as a BIH to stay in close contact with you, and if you have any kind of ideas from individual corporations where we could improve the process, let us know, because we are also a learning entity, and both as a unit and individually, and therefore we need feedback and guidance. But I thank you very much for your feedback and guidance today. I think it was a very interesting lecture for everyone. And I can here also announce the next lecture which we will have. It is planned for the 30th of October, and I think at the moment it's still planned in the current format. Stay healthy, yes, we all have, and stay prudent and careful and follow the advices, because we should not spoil the good coping with the coronavirus pandemic which we had until now. So everyone has to continue being very careful. And with that, thank you very much, and I wish you all a very happy weekend and a healthy summer, and see you soon. Bye bye.
Thank you very much. Bye bye, everybody.